Hope you had an excellent Epic Day, Epic Expo Day. Some interesting things going on around campus. First announcement is that there is a senior seminar today in the biology department here at noon. Pizza provided as always. Arden, Morgan, and Toon will be talking about their senior seminar course on color. Morgan is going to be talking about color reception. Arden, I think Arden's talking about color evolution, communication, and Toon color evolution. So you know something about this, having studied color vision a little bit in the back in the sensory section. Remember cones and opsins. So Morgan will be talking about at least some of that. So that's today at noon in this room. Another announcement, so this is just a Moodle vision, image of the Moodle page for the course. So the key concepts for the practical quiz are up on Moodle. There are some open house options for further studying using the cadaver at these times, which are up on Moodle. If these don't work for you and you would like to ask for an additional open house time, feel free. These are the times that students ask for, which I can accommodate, but that doesn't mean we're limited to these times. The practical quiz process will start a week from today. So this evening, right? So those of you in the Thursday evening section will take your practical quiz a week from this evening. So this evening will be the second of the review things, and then the following week will be the practical quiz. Those of you in the Tuesday section will have your review next Tuesday, and then your practical quiz the following Tuesday. Because of complications both with eCanal and with my own exam, I haven't yet figured out how to make it right in order that some of you are not being forced to take 2 or even 3 exams on the same day. So, but I haven't figured out the options yet. So stay tuned for an email from me with some options. Those of you, basically everybody should be paying attention to this because you might want to take part in the option for the next exam. And those of you in the Tuesday lab, stay tuned for options for the practical quiz. Okay, so I'll figure this out, at least I'll try to figure it out and make an announcement by email probably this weekend. Yeah.
So it's our predicted lab after we did our last lab.
I think so. I have to check. The only, yeah, I think so. So the only reason to, yeah, I think so. Yeah. I'm getting confused about the end of the semester too. It's coming on awful fast. But I'm pretty sure that's correct. The only thing left to do would be to do the course evaluations, which I sometimes do in lab. But since course evaluations is a process that you do, anywhere by computer now, there wouldn't be a reason to allocate time in lab for that.
Do you have another problem set?
No more problem sets. What's left is the practical quiz, exam 3, and the final exam. Those are the assessments that are left. I mean, yeah. I'm not saying it's easy, I'm just saying that's what it is.
And the practical is just like what the same as a normal quiz, right?
Practical is a quiz, same as a quiz, same as a problem set. So at the breaks, those of you who haven't received your quiz 4 back, I have them here. I'll do that at the break. So like bottom line is here, stay tuned for an email about options. Both options if I establish another open house section for the review of the cadaver, and options for taking exam 3 or the practical quiz. Those are the options coming up, and I'll let you know by email. And I'll probably post it to you as well. Okay, so today we're continuing endocrinology and expanding it and to talk about the endocrinology and anatomy and a little bit of genetics and a little bit of physiology related to reproduction, human reproduction. And today we're going to focus on male human reproductive anatomy, physiology, and endocrinology. And then on Tuesday, we will cover female reproductive anatomy, physiology, and endocrinology. And then on Thursday, there will be a review session, question session, to ask questions about the exam coming up the following week. This is our path. We'll get through this. It'll be fine. So, oh my God, there's a giant penis up on the screen. That's what we're talking about here. This is male reproductive anatomy. Let's just talk about it. Right, there's the giant penis up on the screen. So there are some anatomical terms to talk about, to learn, and not just the word penis. There, inside the penis, there is the urethra, which is a tube that carries both urine, in the case of urination, and semen, in the case of reproductive physiology. So the urethra is connected both to the urinary bladder and to a seminal vesicle and the prostate gland. So that's why in the male, the urethra carries both germ cells and semen, that is to say sperm and semen, and urine, depending on which sphincter is open. Inside the penis, there are two main structures that I do want you to know the names of. The corpus cavernosum, illustrated colored, coded sort of pinkish here, and the corpus spongiosum, colored kind of brownish here. These are highly vascularized, spongy tissue, soft tissue, that the vascularization is under control, and we'll talk about that before the end of the class. Increased blood flow and increased blood engorgement of the corpus spongiosum and corpus cavernosum are what's responsible for the male erection. So those are terms to know. The scrotum on the outside, right? The pouch, the scrotum. Inside the scrotum are the testis. So singular is testis, plural is testes. You don't have to memorize that Latinate detail. Testis is where sperm cells are going to be produced, and we'll talk about the physiology of sperm production in a few minutes. And the epididymis, which is a fun word to know how to say, epididymis, is a, well, you'll see, it's a highly tubular structure for transporting the spermatocytes, the sperm cells. Along with the ductus deferens or the vas deferens. So there's two names you'll see in the literature. Ductus deferens and vas deferens is the tube that carries sperm from the epididymis, produced by the testis, to the seminal vesicle, where it is stored for some period of time. The prostate gland is a gland that produces the components of semen that are not sperm. So the prostate gland, and there's some other glands like the bulbourethral gland that help produce semen. But you can cross this one off. That's not on the key concepts list. Prostate, though, is. This here is supposed to be a cross-section of the anus and the rectum and large intestine. That's what this thing is over here is supposed to be. This is the coccyx, the tail bone. Here's the pubic bone in the front. The yellow is supposed to illustrate that there's fat around here. So this is a midline sagittal section. Here is an anterior view really showing the path of the ductus deferens from the epididymis to the, over the bladder, and then over the urinary bladder, and then down into the seminal vesicles behind it. This tube here is the ureter that carries urine from the kidneys to the urinary bladder. So here is a enlarged testis. So here's the testis. Here's the epididymis. These are words to know in terms of the anatomy of the testis. So let's start down here. The surface layer of the testis is called the tunica albuginea, I think. Albuginea. That's the surface of the testis. The testis is divided into lobules by septa. A septum is just a Latin for wall, I think. No, that's a neural. Septum, a divider. So anyway, there are septa dividing the testis into lobules. Each lobule has a set, and I don't think it's just two, but a small number of seminiferous tubules. So the seminiferous tubules are inside the lobule of the testis. The seminiferous tubules from each lobule then merge into this sort of network called the rete testis. It's just a complicated merger of all of these very small seminiferous tubules, which then, so the entrance into the rete testis is the seminiferous tubules, and then the exit is into the efferent ductules of the epididymis. Efferent ductules of the epididymis then sort of merge together to finally form the vas deferens. The vas deferens then is going to go up, loop over the urinary bladder, and then join into the prostate and the seminal vesicle. And so here's the vas deferens. Here's the vas deferens. Okay, so this is male reproductive anatomy to know for the exam.
So, everything that's like here.
Pretty much. Yeah. Yes.
Can you show again where the vas deferens start?
The vas deferens, I think the formal start of the vas deferens is the exit from the epididymis. So this is a tube inside the epididymis, and this is the start of the vas deferens.
All the way down is called the efferent ductules.
I'm not sure what you would call it.
Oh.
So I really am not sure where the formal boundary between the efferent ductules and the vas deferens begins. So I'm not sure what to call this. The beginning of the vas deferens.
If you label that vas deferens, you take off points.
No. If you label this vas deferens, I would take off points. If you label this vas deferens, you'll get absolutely nothing.
Now, I hate to be the guy to ask this, but
Yeah.
I know that like depending on the temperature, testes can either move closer or farther away from the body. Does that have anything to do with this structure?
No.
Or is that just a function of the scrotum?
Nope, it's actually, well, it's more a function of the scrotum than anything else. There's a muscle in here called the cremaster muscle, which is well developed in certain animals and is not particularly well developed in humans, that controls the position of the scrotum. And so cold temperature causes contraction of the cremaster, and the cremaster then contracts and sort of pulls the scrotum up towards the abdominal cavity. So it's a muscle.
Okay, that makes sense.
There's actually another muscle that we'll talk about in a few minutes called the, I'm blanking on it now. Never mind, we'll talk about another muscle in a minute. Bulbospongiosis? Yeah, well, that's another muscle that's important for ejaculation. But the cremaster has, its only function is to raise and lower the testes. Okay, so there are some of these games. You know, we could play these games, but let's not. You're just learning these words. You know, I'll show you. You've seen these games, right? Oh, I thought I had them loaded up. Well, let's load them up. So you can play these games with your friends and laugh and all that. Say, oh my God, look at what I'm learning in school. Right, so you can label these things. Some of them, these games have names that are not on my key list that you may, or that are not required. So there's additional information that is not on the key list, but you can play these games first. So for example, the testes would be here, and the ductus deferens would be here, and the urinary bladder is obviously here. And some of these other names, like urethra was on the list, but ampulla was not. And testes was on the list, but I asked you to cross off bulbourethral. So you can play the game, but focus on the ones that are on the list. And there's another game you can play. Yes, Lauren, question?
Is the ductus deferens just the plural of vas?
It's the, no, it's just a different name for vas deferens. There's another one of these. This is, again, the enlargement of the testes. And so I'm asking you to know the ductus deferens or the vas deferens, and lobule and septum. Those are words that I had up on the screen before. Tunica albuginea, I did, but the head and body of the epididymis is not. That's not a distinction. Epididymis, yes. Head and body, and tail, somewhere. No. Okay. So play the game if you want. It's a way of helping you learn the anatomical terms. The coloring book is also, a good source of information for this. So let's not play the game. I mean, I'm just pointing out that there is the game. Okay. So what we're transitioning now is away from anatomy. Well, we're going to include some anatomy, but we're going to talk about the process of spermatogenesis, the making of sperm. So this is a cartoon version. Let's get oriented. This is supposed to be this is going to be the tubule, the seminiferous tubule. So I'm going to go back up here. No, not that one, this one. Seminiferous tubule is right here. Here's a seminiferous tubule inside a lobule, and now we're looking at it in cross section. So here is the seminiferous tubule. Actually, this whole thing is the seminiferous tubule. This is the lumen of the seminiferous tubule. So this is the seminiferous tubule. And there's another one, and there's another one. So there's more than two in this cartoon, which I think is realistic. And around the seminiferous tubules, so You're gonna have to know that we're gonna talk about the cells of the seminiferous tubule, and now there are interstitial cells called Leydig cells, and that is a name to know. The Leydig cells are the cells around the seminiferous tubules, and they are going to be responsible for responding to a hormone that you've heard of, luteinizing hormone, LH, responding to LH, and producing testosterone. So the Leydig cells respond to LH and make testosterone. Inside the seminiferous tubules is another cell type called the Sertoli cell, which is a name to know. Sertoli cells are in the seminiferous tubule. They are the support cells for spermatogenesis. Sertoli cells respond to another hormone, FSH, follicle stimulating hormone, and promote spermatogenesis. So in this cartoon, this is the outside of the tubule. So this is this surface right here. And this is going through the epithelium of the tubule into the lumen. And what we have are Sertoli cells supporting this structure, Sertoli cells supporting the generation of sperm. So the green cells is the process of spermatogenesis. We're going to talk about that in a minute. Right now, what I want you to know is that Sertoli cells are the support cells of the seminiferous tubules that support, not only make up the tubule, most of it, but they support and stimulate the production of sperm. Sperm cells are not Sertoli cells. Sertoli cells are the cells that support the production of sperm. And they do so in response to another hormone, FSH. Where does LH and FSH come from? Anterior pituitary. What cell type? Gonadotropes. So here is a cartoon from an earlier version of your textbook. Your textbook has a slightly different cartoon, but it's the same information about telling the story of spermatogenesis. Now there's something confusing that all textbooks do, and I want to alert you to this. So the story of spermatogenesis starts at A and ends in sperm here at G. So we're going from A to G. But the story makes it look like these cells are transitioning laterally as well as transversely from the outside to the lumen. So out here is the outside of the seminiferous tubule. So the Leydig cells would be out here, sort of outside in the interstitium. This is the seminiferous tubule. Here's the lumen of the seminiferous tubule. And this is the, the epithelial structure of the seminiferous tubule. Trying to make sure that transitioning from one of these cartoons to another doesn't get you lost.
So what do you need to know from this one?
Well, I haven't told you anything yet. I'm just telling you what is wrong about this picture. What's wrong about this picture is it makes it look like this cell transitions laterally and then divides and transitions laterally and enlarges. That's not what's happening. What's happening is that this cell is moving down in through these spaces between the Sertoli cells. So the spermatocytes, or the developing sperm, are moving through these spaces between the Sertoli cells. They spread it out longitudinally this way because otherwise these pictures would all be on top of each other in one of these spaces. Okay? So there's no lateral movement here. The cells are moving. They're developing, so this cell then develops into two. So now let me tell the story and try to point out where it should be in a cartoon that wasn't on top of each other. I'll transition it over here. So you have, no, not all of you, maybe some of us, have spermatogonia, which are diploid cells on the outside of this epithelium surrounding the seminiferous tubule. These cells divide by mitosis into the primary. So first, the A and B is supposed to be showing a spermatogonia undergoing mitosis. So A cell divides, and now it's 2 cells, and those 2 cells should be right here. The 2 spermatogonia, and this process of mitosis is going on all the time in the male. These 2 spermatogonia, which are here, then undergo more mitosis, but they start to develop into primary spermatocytes. When these cells migrate through this junction between the Sertoli cells, so remember these cells are over here, and now they're starting to move through this tight junction into this space here. So the primary spermatocyte, a primary spermatocyte, is a diploid cell that has gone through this junction, this tight connection between the Sertoli cells of the seminiferous tubule. So the spermatocytes enlarge, the primary spermatocytes enlarge. So here we are at D, an enlarged primary spermatocyte. Remember, this cell over here should be right here, right? This cell divided, and then started to migrate, and then started to enlarge. And so D, which looks like it's way over here, is happening right here. Is that clear? We're moving down this space. D undergoes meiosis, develops into secondary spermatocytes and spermatids, which are haploid. Right? The haploid germ cells of the male are spermatids. So we go from spermatogonia to primary to secondary spermatocytes to spermatids, which then are haploid and develop into these spermatozoa, which develop the flagella, get rid of almost all of their cytoplasm. So they have a small head with very little, just a few mitochondria, very little cytoplasm, haploid DNA, and a flagellum. Israel?
So, in
In D.
is the primary spermatocyte goes through meiosis, and that is what causes it from the secondary.
That's right. And in the end, we have 4 non-identical haploid spermatids from each spermatogonia.
So we have 4 C's like of 1A, right? Like there's 4A
4
and C.
Cs. Well, each A
Right, the divides and divides again.
develops into 2 Cs.
Mhm.
Each of the 2 Cs develop, actually, so A undergoes mitosis, so B is diploid. Diploid cells then develop into spermatocytes, which divide into secondary spermatocytes, which are haploid, which divide in the second step of meiosis into spermatids, which are haploid. I'm not going to test you on the genetics of meiosis. That's something that you've already passed. Those of you who are looking at comps next year should review, but I'm not going to quiz you on meiosis and mitosis. That's CGI, advanced genetics, comps material. Alex.
But how do B and C differ? I was confused about that.
B and C.
Mhm.
In B, we call them B and C are not very different, but what is happening is, so not much difference. So C is, I mean, D is what we're really I guess we're, primary spermatocytes can either be on the outside or on the inside of this tight junction, and maybe that's the difference. Let's not worry about that. The main concept is that spermatogonia develop primary spermatocytes that are diploid, which develop secondary spermatocytes which are haploid, which develop into spermatids. Four non-identical spermatids per spermatogonium per primary spermatocyte. Yeah.
But is this enlargement of the primary spermatocytes happening?
How? Oh, it's, in G phase, I mean, not G phase, gosh, I'm forgetting my own CGI. Yeah, G phase, right, the cells grow and, just make more of everything. Okay? 4 non-identical haploid cells per each starting stem cell, spermatogonia. And here are some pictures that show it sort of, so this is an actual micrograph. Here's a cartoon of what we're seeing. So here's the lumen of the seminiferous tubule. Here are the tails of the sperm cells, the spermatozoa. The spermatozoa stay sort of attached and nurtured by the Sertoli cells for a while. So here the story is shown a little bit more accurately, going from spermatogonium to primary spermatocyte, to secondary spermatocyte, to spermatids, to spermatozoa. The purple thing here, pink thing, is supposed to be a Sertoli cell supporting this process of spermatogenesis. Here is the cross-section of a seminiferous tubule with this spermatozoa on the inside, and these nuclei are probably the nuclei of Sertoli cells, mostly. Well, no, some of them are spermatocytes in the process of developing and migrating through this layer. Remember, let's not forget that outside, in the interstitium, are the Leydig cells that respond to LH to produce testosterone. Testosterone also supports the process of spermatogenesis. We'll talk about endocrinology in just a minute. Yes, Israel.
If you go back to switch to one slide, so that is, all that pink tight junction, that is the,
All this pink are the Sertoli cells. Yeah. And it makes it look like it's one cell. It's not one cell, it's a stack of cells. Here's another blow-up of the seminiferous tubule, Sertoli cell nuclei being identified, spermatids and spermatozoa being identified, Leydig interstitial cells here. They're labeled interstitial cells, but those are the Leydig cells outside of the seminiferous tubule. Here's a scanning electron micrograph. Flagella of the spermatids, spermatozoa, head sort of embedded, being nurtured by a Sertoli cell, something that even the textbook couldn't quite identify. Okay, so now let's talk about endocrinology. GnRH from the hypothalamus, released into the released at the median eminence into the portal circulation. It's carried to the anterior pituitary gonadotrophs. Well, it's carried everywhere, but the LH and FSH receptors, no, the GnRH receptors in the anterior pituitary are on the gonadotrophs. The gonadotrophs have the receptors for GnRH, and they respond to that by making LH and FSH. What kind of LH and FSH are protein hormones. So this is released into the systemic circulation. LH and FSH are co-released by the gonadotropes into the systemic circulation. They go everywhere, but their target is the testis. L8, Leydig cells, the interstitial cells, have the receptor for LH, and they respond to LH by making testosterone. Testosterone gets into the blood and has multiple effects: muscle, brain, bone, but it also is released locally. And this is really a paracrine story, because let's just go back to one of these. Leydig cells are affecting spermatogenesis by local diffusion of testosterone. So testosterone is both a paracrine signaling molecule in the testes and an endocrine molecule, because it goes everywhere and has effects in the brain and muscle and bone as well as in the testes. So this is paracrine signaling from the Leydig cells to the Sertoli cells to stimulate the process of spermatogenesis, to provide growth factors and nutrients and stimuli, probably mechanical stimuli as well as chemical stimuli, to tell those spermatogonium, keep going, keep doing what you're doing, divide, undergo mitosis, meiosis, make more spermatozoa. So testosterone goes everywhere. It has both feed-forward effects, muscle, bone, brain, and it has feedback effects, negative feedback effects. So this is long loop, both of these are long loop negative feedback. It inhibits the hypothalamus from secreting GnRH. This is an effort to maintain the testosterone levels from getting too high. It has negative feedback on the gonadotrophs and tells the gonadotrophs do not make any more LH. So this slide makes a big deal about the fact that testosterone is having negative, long loop negative feedback on the gonadotrophs of the anterior pituitary to tell it to inhibit the production of LH. And then it makes the story that there's another hormone produced by the Sertoli cells called inhibin, which has a primary function for negative feedback, telling the gonadotrophs of the anterior pituitary stop making FSH. The bottom line, but inhibin is not one of the hormones on your key concepts list. So really what you should know is that testosterone has positive feed-forward effects on brain, muscle, bone, but it also has negative feedback effects on the gonadotrophs of the anterior pituitary, and that controls the production of LH. So we have cells that respond to testosterone, both the Sertoli cells and the anterior pituitary cells, and muscle, bone, fat, brain, etc. Yes?
It's positive feedback for testosterone because it's influencing to create more sperm, correct? Or is it just negative feedback?
This is really feed forward.
Feedback.
This is not feedback. This is positive feed forward to that the Leydig cell, the testosterone is telling Sertoli cells to promote spermatogenesis. Just the way that testosterone is telling muscle cells get big. Testosterone is telling hair follicle cells grow or die. Testosterone has multiple effects. Those are all feed forward. The feedback effect is feeding back on the anterior pituitary and the hypothalamus. Yeah.
So now that we know the specific targets for these two hormones, do you want us to specify between FSH and Sertoli cells and Leydig cells? Or is it still
Yes.
just stated there?
No. Now I want you to know that L8, the target of LH cells, is not just the gonads, but is the L8, the Leydig cells in the male, and we'll talk about the specific cells in the female on Tuesday. So now you need to know that the Leydig cells are the cells of the gonad, of the male gonad that respond to LH, and that Sertoli cells are the cells of the male gonad that that respond to FSH. Yes, thank you for making that clear. Any more questions about this feed forward and feed back pathway, culminating really in testosterone and spermatogenesis? So let's just talk about the the hormones now. So LH is a protein hormone. It's released by the gonadotropes of the anterior pituitary. I'm just reinforcing some of the language. In response to GnRH from the hypothalamus. Its target cells are Leydig cells. Its receptor is a GS-linked GPCR. I'm not asking you to memorize that. I am asking you to know the GS story. And its effect is to increase the synthesis and release of testosterone. What kind of hormone is testosterone? Steroid. FSH is a glycoprotein produced by the gonadotropes of the anterior pituitary. It actually has the same, it has two subunits, and one of the subunits is the same as LH. So LH and FSH share an alpha subunit and then have different beta subunits. Stimulus is GnRH. The target are the Sertoli cells. The receptor for FSH is also a GS-linked GPCR. You don't have to know that detail, but yes, you do need to know the GS story. And its effect is spermatogenesis. I'm just pointing out that, I'm repeating myself just because repetition sometimes is helpful. But, this story is then told in, at least in some words, here. Different way of looking at the same information. Testosterone is a steroid hormone. It is one of the androgens. Androgens, including testosterone, are produced from cholesterol, as are all steroids, following, sort of a network of pathways. Will I ask you to know the pathway? No. Will I ask you to know the names of the enzymes? No. But if I give you this map, you ought to be able to figure out a problem related to androgen or estrogen synthesis based on what happens if we, what happens if there's a defect in the P450-17 alpha enzyme. Well, then we're going to make plenty of cortisol and we're going to make plenty of aldosterone, but we're not going to make androgens or estrogens. Yes?
Okay, so I'm still a little bit confused on exactly where things back up if things are damaged. So for example,
yeah,
if P450 aldosterone
synthase.
Has a defect, does that mean it's going to affect everything, even like we're going to have more estradiol as well?
The further you get away from the defect, the less likely that something is going to build up because there's both degradation and synthetic pathways. So there's some answers that are possible but are unlikely, and there are some answers that are definite. So for example, breaking this enzyme, causing a defect in this enzyme, is clearly going to cause a defect in the production of aldosterone. And it's going to cause a buildup of these precursors.
Even though those are still
Oh, I'm sorry, yeah, so if we break this enzyme, we're gonna break all of this. We're gonna build this one up, and that could then lead to an increase in cortisol. But then if we go, so in principle, all of this could back up and increase, but the more steps you have, the less likely it is to actually be detectable as a difference. Yes?
You don't need to know this chart, right?
You need to be able to interpret this chart, but you don't need to know this chart. You do need to know that androgens and estrogens are steroid hormones, and that I guess I will ask you to know that the estrogens are downstream of the androgens. That's something to take away from this figure. You can't make estrogens if you don't make androgens. All of the estrogens all of the precursors of estrogens are androgens. Okay, let's take a break now. We'll come back, we'll talk about testosterone. Testosterone. Steroid hormone produced by the Leydig cells in response to LH from the gonadotropes of the anterior pituitary, stimulated by GnRH from the hypothalamus. It has many target cells, including Sertoli cells, but not limited to Sertoli cells, muscle, bone, brain. The receptor for the steroid hormone testosterone is a cytoplasmic receptor, so it's an intracellular receptor so testosterone diffuses through the membrane, binds to the testosterone receptor in the cytoplasm, conformational change, carried to the nucleus where it acts as a transcription factor for the changes of gene transcription in its target cells, the many target cells. So it stimulates spermatogenesis by stimulates the Sertoli cells to support spermatogenesis, and it has widespread effects on appearance, right? Body hair, right? So that's what this is supposed to illustrate, the production of facial and other body hair, and male behavior. So male phenotype, as well as male behavior. Display, aggression, risk-taking, whatever. This is really aggression. This is sport, but that was the best I could come up with. So maybe this is illustrating increase in body mass, muscle, behavior, not an increase in muscle mass. It's pretty skinny in this picture. Janusha.
For the receptor, now that we know that it's a cytoplasmic receptor, do we also need to know the story? Because we haven't really reviewed.
Which story do you mean?
The intracellular one. It's a very short and the cell phase, but we didn't have to review it in the conference.
Trying to think of what story you're referring to. I mean, binding, conformational change, transport to the nucleus, transcriptional changes. That's the story.
Okay. Okay.
Yes.
What is actually meant by male behavior?
I mean, so I could get on thin ice here, but there is what is I'm not going to tell you what exactly is meant by male behavior. I'm talking about certain stereotypical behaviors of males. And we can think of male animals, doing a male bird doing the display and the dance in order to attract the female. That's sort of what I'm illustrating here in the human. Male behavior in terms of risk-taking. Now, women also do bungee jumping, but males are known to take more risks. That's behavior. Males are so muscle mass increase. That's phenotype. That's body changes. But males have a history of being more aggressive than females. Now, I'm not saying that females can't be aggressive. Of course they can. But there is a pattern of increased aggression by males in response to high levels of testosterone. That's behavior, aggression. that's not aggression, that's sport, but muscle. Risk-taking, aggression, display behavior. Those are behaviors. So, there's an interesting story about the development of the male genitalia, which I'm not going to test you on. So this is just an interesting factoid that you can take away from A and P, and that is that on the Y chromosome, remember Y is what defines a male, on the Y chromosome is a gene called the SRY gene, which makes the SRY protein. Some of you may remember this because you wrote a poster about this back in CGI. So the SRY protein is key for the development of testes and key for causing the degeneration of a female structure. So by default, we are female. And then, but if we happen to have the Y chromosome and we make the SRY protein, we become male by promoting the development of testes and inhibiting the production of female Mullerian ductwork. So testosterone, along with the SRY protein, development of male structure and development of male reproductive genitalia. Now, there's some interesting things that come out of abnormalities in this. Someone can be genotypically male, but if there's a defect in the SRY gene or a defect in the SRY protein, they're genotypically male, but they might be phenotypically mixed or female. There's a really interesting story about a neurobiologist. So I happen to read a story about a neuroscientist. He was a very famous neuroscientist. He worked on glial cells, which I don't teach very much about in this course, but glia are the other major cell of the nervous system, besides neurons. So glia are really important, but I don't cover them very much. Anyway, he became a gliobiologist. And his name what? He died not too long ago of pancreatic cancer. He was a famous scientist. His name is Ben Barres, but he was born Barbara Barres. He was born phenotypically female, but not exactly phenotypically female. He did not have a uterus. So there was abnormalities in the development of this person. But the external genitalia were female, so his parents named her Barbara. But then, as you can tell, she transitioned as a scientist, as an adult, to become a male, a very famous male. And in fact, Barbara Barres, was pretty famous even before she transitioned to becoming Ben. And so, and he wrote a very interesting autobiography called The Autobiography of a Neuroscientist or something like that. And it goes into great detail about his discoveries in gliobiology. So if you're interested in gliobiology, it's a pretty interesting book because he made some major advances. But he also talks about his being raised as a girl, uncomfortable with that, unhappy with being treated like a girl when in graduate school. A pretty famous story where he overheard somebody talking about one of his presentations after he transitioned. He overheard this person say, Ben is does so much better work than his sister. He doesn't have a sister. He was referring to this person wasn't aware that he was talking about the same person. So, interesting story, interesting life. His mother died of breast cancer, so he had the BRCA2 abnormality that gave him the predisposition to breast cancer. So he had his mammary glands were removed, but it also predisposed him to pancreatic cancer. And that's what killed him. So an interesting story about some of the nuances, right? I teach male and female, but I want to make it clear, it's not, we're not binary. There are nuanced parts of the male-female story. Yes?
So, I mean, I know that the story is more complicated than just down to the SOI gene, but if we are by default female, how can someone be genotypically female but phenotypically male?
Does that happen? Is someone with
Isn't
an
that what
X?
you said about the
No, I said that someone can be genotypically male and phenotypically female.
Yes, what about Ben Harris? Did
Ben?
you say he's genotypically female, but
I-
phenotypically male?
He did not reveal the story of his
Oh,
genotype,
so
so I don't
okay,
know.
I caught
He is
it.
not necessarily this story.
Okay. Yeah, just sort of, yeah.
I'm not sure. He had unusual phenotype that made it actually rather easier for him to transition. I mean, he didn't have a uterus. He wasn't producing as much estrogen as, I don't think he had ovaries. So he did have breasts. I mean, we all have breasts, but he had phenotypically female breasts until he transitioned. Okay, so now let's talk about male reproductive physiology, the erection and the ejaculation. Again, this will not be on a test. This is just an interesting information to walk away from with this class. So, erection starts in the brain, arousal. It activates the parasympathetic nervous system. That is going to be on a test somewhere again, right? That will be on the final. The target of this, so activation of the parasympathetic nervous system activates the endothelium of the corpus cavernosum and corpus spongiosum. That will be, so that part will be on the test, right? I told you that was on the test. The parasympathetic nervous system activates the endothelium surrounding the vasculature of the corpus spongiosum and corpus cavernosum, causes the release, or causes the activation of guanylyl cyclase in smooth muscle. Guanylyl cyclase, increase in cyclic GMP. Cyclic GMP causes relaxation of the vascular smooth muscle, which leads to increased blood flow and the blood engorgement of the corpus cavernosum and corpus spongiosum, which leads to the erection. So there's the story of the erection. Cyclic GMP is produced by guanylyl cyclase in response to nitric oxide from the endothelium. So this is a paracrine signaling molecule that activates an enzyme that increases, what do we call this class of molecule? I'm just sort of starting to remind you of some words that you knew from way back. Second messenger. So ACH is a neurotransmitter, nitric oxide is a paracrine signaling molecule, guanylyl cyclase is an enzyme that makes a second messenger that causes relaxation of smooth muscle. Cyclic GMP is broken down by an enzyme called phosphodiesterase. There's an enzyme name you know, phosphodiesterase 5. It's not the same phosphodiesterase that breaks down cyclic G in your In your retina. It's a different phosphodiesterase. What do you suppose? This is the target of a drug that you think of. What would inhibiting phosphodiesterase 5, not phosphodiesterase 1, thankfully, phosphodiesterase 5 specifically, what would that enzyme do? Prolong or increase erection? What do we call that drug? Viagra. Viagra was discovered. Interesting story about Viagra. They were not searching for a treatment of erectile dysfunction. They were looking for a treatment for male pattern baldness. and they came across a side effect, which turned out to be a pretty lucrative side effect. Anyway, Viagra and the other treatments for male erectile dysfunction work by inhibiting the enzyme that breaks down cyclic GMP. Corpus cavernosum, corpus spongiosum, erection. Ejaculation is not due to the parasympathetic nervous system, it's actually a process triggered by the sympathetic nervous system. There's a nerve and a muscle. That was the name of the muscle I was trying to come up with before, bulbospongiosus muscle. It's a muscle that undergoes a spasm. It's a spastic contraction of this muscle, which leads to ejaculation of the semen and sperm from the seminal vesicles and the prostate.
Where is this muscle?
Pardon?
Where is it?
Where is the bulbospongiosus muscle? So, it is, so let me see if it's shown even schematically in any of the cartoons. I mean, schematically, it's around here. So contraction, it's sort of, yeah, it's around here, and contraction in this area leads to squeezing of the chambers that are storing sperm and semen. I think it's a muscle that sort of wraps around here. The anatomy coloring book might have a pretty decent schematic image of where this bulbospongiosus muscle is. And I do think bulbospongiosis, I think, is on the key concepts list now. But I would not ask you to identify, I would ask you to describe its physiology, but its anatomy is a little fuzzy. Okay, let's do some quickers. Let's just finish up with a couple. See how well this sunk in. Which of the following are part of the pathway that leads to the secretion of testosterone? So I'm asking about the feed-forward pathway to the production of testosterone. A and C are the best answers because the hypothalamus releases GnRH, the anterior pituitary releases LH. The target is not the Sertoli cells. What would be included here? Leydig cells. Which of the following are parts of the pathway leading to development of spermatocytes? This A, B, and D. Well, all of them. Why would it be all of them? LH makes, Leydig cells makes testosterone. Testosterone is the paracrine signaling to the Sertoli cells to stimulate spermatogenesis. So all of these are correct. Okay, so here's one of those problems. The precursors to testosterone are the same as the precursors to cholesterol. What would happen to a testosterone levels if cortisol synthesis was impaired due to a defect in the P450C21? So, do I expect you to know this? No. Without referring to the figure? No, but if you look at slide 13, you should be able to figure this out. What would happen to testosterone levels if cortisol synthesis was impaired due to defect in P450C21? So let's look, it's too small to read. P450C21. Well, let's go back and find the figure. P450C21. P450C21. P450, C21. So we're breaking both of these pathways. What's going to happen to testosterone? It's going to increase. And finally, I'll tell you another interesting story of male reproductive anatomy. So let's read this abstract from a case report that was published in the 70s, 1979. This is a case report, a single person. In 2 separate procedures, a psychiatric patient performed a bilateral orchiectomy on himself and then later attempted to denervate his adrenal glands. This case suggests that physicians should be alerted to the possibility of self-surgery occurring in patients who have sought elective surgery and have been rejected. So there's some backstory here. This was a patient who knew he was a psychiatric patient. He had been committed to a psychiatric ward and had been released and committed. He had a history of psychiatric disorder. Schizophrenia, which was controlled and then not controlled. You know, he oscillated in the level of the schizophrenic, the control of his schizophrenia. What's a bilateral orchiectomy? Bilateral, 2, cutting, ectomy. The orchid is a Latin name for the testes. So this is castration. Bilateral orchiectomy is castration. First performed a bilateral orchiectomy on himself. So this is a person who castrated himself surgically. He sterilized his scrotum and got a scalpel. He was a medical student, actually, so he had access to tools, and he anesthetized him. I mean, he did a local anesthetic and sterile technique and everything, and removed his own testicles. Why? He knew he had a problem with reality, I mean schizophrenia, and he knew that testosterone was associated with aggression, and he was afraid of doing something terrible. So he went to a psychiatrist and said, I'm afraid of hurting somebody because of my condition. I want you to castrate me. And they said, you can't do that. And so he did it to himself. So that's what this physician should be alerted to the possibility of self-surgery in the case of people who have asked for it and been rejected because you just don't do that. So now, why did he attempt to, so that's not a hard surgery to do. A bilateral orchiectomy is not an extremely hard procedure. But denervating his adrenal glands is a hard procedure, right? The adrenals are here. It's not only, I mean, it's hard to reach, right? And then, you know, cutting the nerve that innervates the adrenal without cutting something important like a blood vessel is actually pretty hard. And that's what he did, in fact. He did the surgery on himself, and he cut a blood vessel, and he then he called the ambulance because he knew he was bleeding to death, and they took him to the hospital, and that's when this story, you know, that's when this came up, when the physicians realized, this isn't it, what's going on here? So why would he attempt to denervate his adrenal? Why, now I'm going to put this up, why would he attempt to denervate his adrenal? Because he knew, as a medical student, that the adrenal gland, the adrenal cortex, in addition to producing cortisol and aldosterone, also produces small amounts of androgen. So he said, I want to make sure that I don't hurt somebody, I'm going to get rid of all the testosterone, and so not only am I going to remove the testes, I'm going to denervate the adrenal cortex, the adrenal, so that I don't make testosterone here in the adrenal gland. because the adrenal is a small source of testosterone.
Wow.
It didn't work. I mean, he almost died because but so how did you do this? He had mirrors, and he had some sort of a special tool that he developed to make cuts in his own back. Oh, in fact, there's a part of the story. He didn't call the ambulance. He called his friends and said, "I've made a mistake here. Can you stitch me up, please?" And that's when the friends called the ambulance. Question? Do you have a question?
So what if, like, hypothetically, if he did cut off the adrenal cortex, what if there be like an addition or like a surplus of like testosterone to?
If he denervated the adrenal, he's going to I'm not sure it makes a ton of sense, because denervating the adrenal is not going to prevent the, what is the stimulus for production of testosterone? He's still going to have his anterior pituitary making LH and FSH. The LH is, but he doesn't have any more, these cells are making testosterone. The adrenal gland is making testosterone not in response to LH and FSH, just sort of because there are the enzymes there. And it doesn't make very much of it. So the idea is we're going to cut off the adrenal innervation in order to tell the adrenal just stop making everything. Yeah.
So if he did that, when he died, he was really close.
Yeah. So that's a good point. Apparently, maybe he's right that the adrenal gland, even if you denervate, so he didn't try to remove his adrenal glands. He tried to denervate it. And so maybe he's saying the adrenal glands are going to just sort of constitutively make enough cortisol to survive, but not enough testosterone to affect my baby. I'm not sure it's logically, he's a psychiatric, he's schizophrenic, he's crazy. I wonder, so 1979, so he was 20 or so, let's say he was 25 in 1979, so he's my age. I don't know. Okay, so some of you I'll see in lab this evening. We'll do another review. And stay tuned for information about the practical